Okay, let's really dig into this. You've given us some really fascinating sources here, taking us from the ground level, what cancer actually is, all the way to this very specific and kind of surprising idea of cancer stem cells.
That's right. And cancer, it often feels like this huge, overwhelming
Oh,
topic.
totally.
But this material, it actually breaks it down quite nicely.
Hmm.
First, it makes clear that cancer isn't, well, it's not just one thing. It's a group of diseases,
Right.
right? All sharing certain characteristics.
Okay.
And then it introduces this cancer stem cell idea, which, as you said, is surprising, but it helps explain a lot, like why treatment is so difficult sometimes and maybe where things are headed in the future.
Exactly. So our plan here is to go through this material with you, sort of pull out the key nuggets, the really important bits, and hopefully give you those aha moments. We want to make it clear, engaging, without you drowning in, all the jargon.
Sounds good.
Yeah.
So where does the material start? With the basics, the definition. What actually defines this group of diseases we call cancer?
Right. It comes down to uncontrolled cell behavior. The sources really highlight three main things.
Okay.
One, cells just growing without limit, basically ignoring the stop signals.
Yeah.
Two, they invade nearby tissues. They push into places they shouldn't be. And three, crucially, they can spread. Break off, travel through the bloodstream or lymph system, and set up shop somewhere else, form new tumors. That's metastasis.
And the sources also point out that where the cancer starts really matters.
Ah, yeah, the types.
Yeah. Carcinomas from epithelial tissues like skin,
Mm.
linings of organs,
Mhm.
sarcomas from connective tissues, bone, muscle, adenocarcinomas from glands.
Mm-hmm.
And, their origin affects how they act, how they're treated. So context is key right from the start.
And here's maybe the most fundamental point. It's hammered home in the sources. Cancer, at its absolute core, is a genetic disease. It's all about changes, mutations in the DNA.
Right, pathological changes. And these mutations, they can come from different places.
Yeah.
External factors, maybe?
Yeah, things like UV light, certain chemicals, the stuff we call carcinogens, they damage the DNA.
But it's not always external, is it?
No, exactly. Sometimes errors just happen when cells divide. Our cells copy trillions of DNA letters all the time, and occasionally mistakes slip through, despite our repair systems.
And importantly, for most cancers, we're talking somatic mutations,
Meaning?
meaning they happen in your body cells during your lifetime. They're not usually in the egg or sperm, so you don't typically inherit them and you don't pass them on. Acquired changes.
Got it. And these changes can be, well, pretty varied, right? Tiny little typos in the DNA code
Or huge rearrangements, whole chunks of chromosomes deleted, swapped around, duplicated. A real mess sometimes.
And it's usually not just one bad mutation. The material stresses that cancer development carcinogenesis is a multi-step process.
Right. It takes a buildup, an accumulation of mutations over time, usually in one single cell lineage. We all collect mutations as we age. That's normal.
But cancer happens when a cell hits a kind of uh, unlucky lottery, gets that specific combination of critical mutations.
Exactly. Which leads us to the difference between benign and malignant tumors. Because tumor just means an abnormal lump, right?
Yeah. Benign ones are, well, relatively contained. They grow, but they don't spread. They stay put.
Malignant tumors, though,
Yeah.
that's cancer. They're not contained. They invade. And crucially, they metastasize. They spread.
And that process, metastasis, your sources really emphasize how complex and, frankly, how dangerous it is. It's not easy for a cancer cell.
No, it's a whole obstacle course. The cell has to break away, get into a blood vessel or lymph vessel that's intravasation.
Survive floating around in the circulation, which apparently most don't survive.
That's right. It's a hostile environment. Then, if it survives, it has to get out of the vessel somewhere else, extravasation. Then form a tiny little outpost, a micrometastasis.
And then actually manage to grow into a proper secondary tumor, colonization.
It's incredibly inefficient, really. But the few cells that manage that whole journey, they're the reason why cancer is often fatal. Understanding metastasis is just vital.
And driving this whole process of mutation and spread is often something called genetic instability.
Right. It's not just that cancer cells have mutations. It's that their whole system for maintaining DNA integrity gets, well, broken. They become mutation prone.
So they start accumulating more mutations faster.
Exactly. Defects in DNA repair, problems with cell cycle checkpoints, It creates a vicious cycle.
Which feeds into this idea of tumor evolution. It's like natural selection, but inside the body.
That's a great way to put it.
Yeah.
A cell gets a mutation, giving it an edge. Maybe it grows faster or ignores a stop signal better.
So its descendants take over that little niche.
Right. Then, within that population, another cell gets another advantageous mutation. Maybe it can attract blood vessels better.
And its descendants outcompete the others.
Exactly. Round after round of mutation and selection. Survival of the fittest, or maybe nastiest cell, and the tumor gets progressively more aggressive, more resistant over time. Explains why treatment can become less effective.
And all these abilities they acquire, the sources bundle them up as the hallmarks of cancer. These are like the key upgrades cancer cells need to thrive.
Yeah, the essential toolkit for being a successful cancer cell.
Things like making their own go signals.
Yep. Sustaining proliferative signaling. Ignoring the stop signals, evading growth suppressors.
Resisting cell death apoptosis, like disabling that P53 protein you mentioned.
Exactly, the guardian of the genome. Also, becoming immortal, limitless replication, often involving something called telomerase.
Getting their own blood supply angiogenesis.
Crucial. And of course, the ability we just discussed, invasion and metastasis. Gaining these hallmarks gives them that massive competitive edge over normal cells.
Okay, so that paints a picture of cancer. Genetic, evolving, defined by these hallmarks, but then the sources throw in a curveball. This idea that, wait a minute, maybe not all cancer cells within a tumor are actually driving the process equally.
Exactly. This is where cancer stem cells, or CSCs, come in. It started with observations from experiments that were, frankly, pretty surprising at the time.
What kind of experiments?
Well, researchers would take a tumor, break it down into individual cells, and then try to grow a new tumor by injecting those cells into, say, immunodeficient mice.
Okay, and what happened?
They found that you often needed to inject a lot of cells to get a new tumor to grow. Sometimes only a very small percentage of the injected cells were actually capable of initiating that new tumor.
Huh, not every cell could do it.
No. The material gives an example, like maybe for a particular breast cancer, you'd need to inject tens of thousands of bulk tumor cells, but maybe only a couple of hundred specific cells could actually kickstart a new tumor.
Wow, okay. So that implies the tumor isn't just a uniform blob of identical cancer cells.
Precisely. It implies heterogeneity, different cell types within the same tumor. And only a select few, this rare population, seem to possess this tumor-initiating power. These were dubbed cancer stem cells.
Because they act like the stem or origin of the tumor's growth?
Kind of. It led to this hypothesis.
Yeah.
Maybe tumors are organized hierarchically. Similar to how normal tissues have stem cells that generate all the specialized cells, maybe tumors have their own stem cells driving their growth and diversity.
Okay, so what defines these CSCs according to the hypothesis and the sources?
Well, first and foremost, they are defined by that ability. They're the only cells within the tumor proven to be able to initiate a new tumor when transplanted. They're the seed.
Got it. And like normal stem cells.
They can self-renew. They can divide to make more CSCs, maintaining their own little pool.
But they don't just make copies of themselves, right?
No, that's the other key part. They are multipotent. They can also differentiate, giving rise to the other non-stem cancer cells that form the bulk of the tumor mass. That's where the heterogeneity comes from. The CSCs generate the diversity.
Okay, so tumor initiating, self-renewing, differentiating. What else?
They're typically a very small fraction of the total tumor cells. Maybe less than 1%, sometimes more, it varies. They possess that limitless replicative potential, that immortality hallmark. And critically for treatment,
They're tough.
they are often highly resistant to conventional therapies like chemotherapy and radiation.
It's interesting that comparison to normal stem cells. Normal stem cells maintain our tissues, right? Controlled division, differentiation.
Exactly, they're essential. CSCs seem to hijack those normal stem cell properties, self-renewal, differentiation, but twist them for uncontrolled growth and creating this malignant structure.
Where do these CSCs even come from, do we know?
That's still debated, but the leading theory, mentioned in your sources, is that they arise from normal tissue stem cells that undergo cancerous mutations. They already have the self-renewal machinery, it just gets corrupted.
Makes sense. Any other possibilities?
Yeah, maybe progenitor cells that are slightly more differentiated than stem cells could get mutated and somehow de-differentiate or gain stem-like abilities?
Or even existing cancer cells.
Potentially. Some suggest that regular cancer cells might undergo changes, like this process called EMT, epithelial mesenchymal transition, which can give them more mobile stem-like features. They might reactivate genes usually only active in embryonic stem cells like ULT4 or NANOG, sort of reverting to an earlier state.
Okay, so if these CSCs are the ones starting tumors, keeping them going, creating the different cell types, and they're resistant, the clinical implications must be huge.
Absolutely massive. Your sources spell it out. CSCs are thought to be the main triggers of metastasis. They're the cells that likely survive therapy. And because they survive,
They cause the cancer to come back, relapse.
exactly, they're seen as the persistent root of the cancer. If you don't get the root, the problem returns.
And this helps explain why chemo, even if it shrinks a tumor way down,
Often doesn't lead to a cure. Because traditional chemo and radiation, they're designed primarily to kill cells that are dividing rapidly.
which is most of the tumor bulk, right? The non-stem cancer cells generated by the CSCs.
Precisely. But the CSCs themselves, they might be dividing very slowly or even be dormant, quiescent, just kind of sitting there.
So the chemo just flies right past them,
Well,
essentially.
they're just not susceptible in that state. Plus, they seem to have intrinsic defense mechanisms.
Like what?
Your sources mentioned several, those drug efflux pumps proteins in their membranes, like certain ABC transporters, that literally pump chemotherapy drugs back out of the cell.
Wow, like tiny bouncers kicking the drugs out before they work?
Pretty much.
Right.
They also often have beefed up DNA repair systems, so they're better at fixing the damage caused by chemo or radiation. And they seem inherently more resistant to undergoing apoptosis, that programmed cell death signal.
There's that analogy in the sources, the dandelion one. I thought that was powerful.
Yeah, it really captures it. Chemo is like mowing the lawn.
Mm-hmm.
You cut off the dandelion leaves, the bulk tumor shrinks, and it looks good for a while.
But you didn't get the root.
Exactly. You didn't kill the CSC root. So inevitably, it grows back. That's relapse.
So understanding this completely changes the goal of therapy, doesn't it?
It really does. The focus shifts. It's not just about shrinking the visible tumor anymore. The ultimate goal becomes specifically targeting and eliminating those CSCs.
The idea being if you kill the root,
Yeah.
then the rest of the tumor, the bulk cells that can't self-renew or initiate new tumors, they might eventually die off on their own or maybe become easier targets for other therapies or the immune system. You're aiming for a lasting cure by preventing the recurrence.
Okay, but if these CSCs are rare and tricky, how do scientists even study them to figure out how to target them?
Yeah, that's a challenge. You need ways to isolate them first. The sources mention a few methods.
Like,
One is called the side population, or SP method.
Hmm.
It exploits those efflux pumps we mentioned. You treat the cells with a special dye, and the CSCs pump it out.
right, so they look different, they fall into a side population when analyzed in a machine called a flow cytometer.
Clever. What else?
You can try to grow them in specific lab conditions, special media without serum, often on plates where they can't stick down. CSCs tend to survive and grow as these little floating spheres called tumor spheres or spheroids.
Ah, okay. Kind of selecting for them based on their growth properties.
Exactly. And then there's using cell surface markers, identifying proteins that are preferentially found on the surface of CSCs in a particular cancer type.
Like antibodies against proteins like CD44 or CD133?
Yes, those are common examples. You use antibodies tagged with fluorescence to label the cells carrying the marker and then physically sort them out. Though finding reliable universal CSC markers is still tricky.
Okay, so you've isolated some cells you think are CSCs. How do you prove it?
That's where the gold standard assay comes in, the tumor genicity assay.
The mouse test again.
Yep. You take your purified cells and inject them into those immunodeficient mice. If they form a tumor, especially if they can do it with very few cells injected compared to unpurified cells, that confirms their tumor-initiating ability.
And what about proving self-renewal?
For that, you do serial transplantation. You take the tumor that grew in the first mouse, isolate the suspected CSCs from it, and inject them into a second mouse.
And repeat.
Repeat. If you can show that these cells can consistently regenerate the tumor over multiple passages, multiple mice, that's strong evidence for true self-renewal capacity. It confirms they are the persistent root population.
Okay, so we can find them, we can test them. Now, how do we actually target them therapeutically? What strategies are being explored?
Well, since they're seen as the key drivers of the whole mess initiation, spread, recurrence hitting them is critical. Your sources outline several approaches.
Like going after their internal wiring.
Exactly. Targeting the specific signaling pathways that CSCs rely on for self-renewal and survival. Things like the WANT pathway, Hedgehog, Notch. These are often hyperactive in CSCs.
So the idea is to block those signals.
Right. Force them to differentiate into less harmful cells or maybe trigger their death. using drugs or even things like gene silencing techniques.
What about attacking them from the outside, using those surface markers?
That's another major strategy, developing antibodies that specifically bind to CSC markers like CD133 or CD44. These antibodies could directly kill the CSCs or flag them for destruction by the immune system.
Or even be used to deliver toxins directly to the CSCs.
Precisely. And this ties into things like CAR T-cell therapy, where you engineer a patient's own immune cells to recognize and kill cells bearing a specific CSC marker.
And we talked about their resistance. Can we tackle that?
Absolutely. A key goal is overcoming that drug resistance, especially developing inhibitors for those ABC transporter pumps.
So the chemo drugs stay inside the CSCs and can actually work.
Exactly. Making them sensitive to conventional therapies again. Combining pump inhibitors with standard chemo is a big area of interest.
What about their hiding place, the tumor microenvironment?
That's crucial too. CSCs don't exist in isolation. They rely on a supportive niche within the tumor. So strategies aim to disrupt that niche.
Oh.
Things like anti-angiogenesis therapies to cut off their blood supply, therapies targeting the low oxygen conditions they thrive in, or blocking signals they get from surrounding cells like certain chemokines or interactions with immune cells like TAMs, tumor-associated macrophages.
And using the immune system itself.
Yes, harnessing the immune system to recognize and eliminate CSCs is a huge focus. Checkpoint inhibitors, therapeutic vaccines designed to target CSC antigens, ways to boost natural killer cells or T cells to go after CSCs.
It seems like a multi-pronged attack is needed.
Very much so. Other approaches mentioned include using nanoparticles for more targeted drug delivery specifically to CSCs, and even using other types of stem cells, like mesenchymal stem cells, which naturally home to tumors, as delivery vehicles for anti-CSC therapies.
Wow. Okay, so we've really covered a lot of ground. Started with cancer basics, the genetics, the evolution, the hallmarks, the danger of metastasis.
Then pivoted to this really paradigm-shifting idea of cancer stem cells, this minority population that acts as the engine, the root, explaining resistance and relapse.
And that led us to the future, really. How understanding CSCs is driving the search for new therapies that don't just shrink the tumor, but aim to eradicate that root, to achieve a lasting cure.
So wrapping it up, what's the big picture here? Your sources paint cancer as this incredibly dynamic, complex, evolving adversary. But this CSC model, it gives us a new lens, a framework to understand why it's so resilient.
It shifts the focus, right, from just attacking the rapidly growing cells
To specifically identifying and eliminating those rare, hardy cells that can actually restart the whole disease. It's a fundamental shift in strategy.
Okay, let's leave you with a final thought to chew on, something that emerges from all this. If these cancer stem cells share so many properties with our normal essential tissue stem cells, self-renewal differentiation pathways, how incredibly difficult must it be to design therapies that selectively kill the bad stem cells, the CSCs, without wiping out the good stem cells that our bodies absolutely need for normal function and repair?
That is the central challenge, isn't it? Finding that therapeutic window. It highlights the immense precision needed for these next generation treatments. It's where a lot of the really intense research is focused right now.